Good morning, everyone. Thank you very much for tuning in. So, as promised, today we are going to have our synthesis. Now, what I did is I chose several topics in microbiology. Some of this we already discussed. You will have this in your module. But I just would like to highlight some concepts that I think it's very important for you to learn. So being in a microbiology class, it is very important to learn principles that you might be able to use in the future, for example, in research or in higher courses that you have. So I hope you are ready. I can see that there are still 15 tuning in. Let's wait a bit for others to be in so they won't miss our lecture. Oh, good morning to our Dean, Dr. Hermini Palya. He's in. Let's give others 3 minutes. So let's start at exactly 9:35. Good morning, everyone. There I can see all your names. So what I'm going to do is to start print screen so I won't miss your attendance. Paki-remind naman yung iba. Kamo, we're gonna start soon. Oh, by the way, while waiting, if you are with your module, you can also try and have a look at it and make a list of questions that you would like to ask. Baka meron kayong mga topics na hindi masyadong naintindihan. Pwede nyong i-type ang question sa ating chat para pwede nating ma-address later. It's either during the lecture or after the lecture. My lecture would probably last 45 minutes to 1 hour. Kung medyo bibilisan, baka mas maiksi ng konti. But let's see. Anyway, this will be our final lecture for this semester. I'm just really hoping that at least you got something from this course. O trying hard na talaga si ma'am para lang may matutunan tayo. Kasi nga with our situation now in this pandemic, nakakapanibago pa rin that we do not have face-to-face interaction. Sanay ang mga teachers na magturo with the students in front of them. But like this, like I'm teaching you and you are chatting with me, di ba parang this is something new for all of us. But sige lang, we are survivors. So we can do this. The reason why I arranged for this synthesis is sa sobrang dami ng module na ginawa ninyo for the whole semester, I'm wondering how much you have retained, how much you have understood of all the topics. Kaya gusto ko mag-highlight para kahit papano merong matira sa inyo. Is that fair enough? Okay ba yon? So it's 9:35. I'm gonna start my lecture. So for those who will be coming in late, just type your name so I can still check your attendance later. And for those who would like to ask question, feel free to ask question. Just type your questions and I will answer at the end. All right. So this is a synthesis of microbiology. So let's start by defining prokaryotes. Microorganisms such as bacteria are actually member of the prokaryotic group. That means that they do not have nucleus. And their genetic material is not stored in nucleus, but instead it is called nucleoid. So what's the difference? The nucleoid is not bound by a membrane, unlike the nucleus that it's bound by the nuclear membrane. The nucleoid is the region of the DNA concentration. That is where the genetic materials are present. So it's just the same. Prokaryotes, eukaryotes, the genetic material is still the DNA. And prokaryotes do only have fewer organelles compared with the eukaryotes. So they are less complicated. But then, that less complicated, as I have given example, the sikat na Escherichia coli, E. coli has approximately 3,000 genes governing specific traits. So kung isang cell lang siya, di ba, unicellular nga, but it contains a lot of genes already, how much more the eukaryotes and how much more the multicellular organisms just like us. And remember this, all bacteria are prokaryotes. So, what are the different parts of prokaryotic cell? So, we have this also in your module. I just would like to highlight a few. For example, here you have the pili. And then you also have the fimbriae. Actually, this one, yung short extensions are called the fimbriae. They are used for attachment to surfaces. And then, of course, there are ribosomes suspended in the cytoplasm. The function of the ribosomes in prokaryotic cell is just the same in eukaryotic cell, that it makes the proteins. So remember that. Capsule. The capsule is very important for bacteria because it determines the virulence of the microorganism. Usually, when it is capsulated, it is pathogenic. What does that mean? Pathogenic means that they are capable of causing disease to the host. Yan. So, capsule. And also, it prevents from being easily digested, easily affected by, for example, the antibiotic. No. And then another is the cell wall. Similar to plants, bacteria also do have cell wall as a means of protection and what determines its shape as well. And then the plasma membrane, you also have here the nucleoid, you already mentioned, and the flagella. It's, in layman's term, it's a swimming organ. It's the motility organ of the bacterial cell. So these are the important parts that you should know about the prokaryotes, specifically the bacteria. Now let us go into the principles of bacterial growth. So bacteria grow and increase in number by binary fission. What does that mean? 1 becomes 2, 2 becomes 4, and so on and so forth. So in shorter time, we call it the generation time. When one cell divides into 2 cells, the time that it takes for one cell to divide is what we call the generation time. For some microorganisms, for some bacteria, the generation time is a lot shorter. And for others, it can be longer. So generation time can be 10 minutes, 15 minutes, 30 minutes, 1 hour, 1 day. But for microorganisms, really, they can increase in number exponentially. Kasi nga, diba, they double every generation time. So that, if you leave your food outside unrefrigerated, tapos the temperature pa is a little warm, kasi we, here in Palawan, here in the Philippines, our weather is really warm naman talaga. The microorganisms are given the very good environment to grow. Kaya pag iniwan mo yung pagkain dyan, mamaya mo lang panis na. That means that microorganisms are already proliferating. So madami na sila doon that are consuming the food. And it changes the quality, producing the acid as manifestation of fermentation. And also, dyan nangyayari yung mga food poisoning kasi some of those microorganisms affecting are producing toxin. So, nakaka-cause sila ng sakit ng tiyan, pagsusuka, no? The food poisoning symptoms. Now, microorganisms also grow naturally in the environment. In the history, microorganisms are just studied in the laboratory. Na-imagine ko yung mga scientists dati, no? How they study microorganisms. But as the environment, the climate, and everything change, microorganisms also adapt. And they also change. So, one of those is actually being able to survive. So, parang kanilang adaptations for them to survive. And one of the ways is for them to live as a community. So, maramihan, no, as a community, and that is termed as biofilm. Ano, do you wonder kung bakit minsan yung surface ng rock madulas, o kaya yung surface ng toilet bowl natin pag hindi nalilinis, parang nagiging slippery, mayroon pang mga pinkish-pinkish, greenish-greenish na mga color. Tapos yung sink natin pag hindi nalilinis, ganun din, parang slimy siya. That is because of the formation of the biofilm. But one of the most important applications of this biofilm is antibiotic resistance. Kasi kapag magkakasama-sama na sila, minsan ang hirap na nilang i-treat with antibiotics because they develop a mechanism of protection. Kagaya nito, parang meron na silang balot. So, dati-dati, tig-iisa lang sila. Pagkatapos, nagsama-sama sila, and then dumami. Kasi syempre magre-reproduce, diba? And then dumami ng dumami, and they secrete this so-called extracellular polymeric substance. And that protects them. So, itong biofilm, isa sa mga siguro makaka-relate kayo na example is yung tinatawag nating dental plaque. Yan, no? Diba? Pag hindi kayo nagtutoothbrush ng ngipin nyo regularly, when I say regularly, at least 2 to 3 times a day, makikita mo na magkakaroon ng plaque, no? May kulay sa ngipin, o kaya parang magkakaroon siya ng itim, eventually magkakaroon ng cavity, tapos ng cavity, sasakit na yung ngipin, bubunutin na. So, ganun siya, no? That's an example of the application of the biofilm na characteristic ng bacteria. So, if we want to study microorganisms singly, yung siya lang, walang kasamang iba, kasi di ba usually they exist in the environment with many other microorganisms, paano natin gagawin? So, we have to obtain pure culture. Isa lang, pure culture. And we can grow these microorganisms either in liquid or solid culture media. The broth or the agar media. No, marami tayong klase-klase, no, meron tayong, mamaya matatackle natin yan. But, microorganisms are forming, those which are culturable are forming colonies. Kagaya dito sa picture na nakikita nyo, itong mga bilog-bilog, these are microbial colonies or bacterial colonies. So, pwede nating ma-pick yung colony kasi isang colony, it means isang pure microorganism. Yan, isang klase lang sila. So, you can pick a colony and then subculture it in other culture medium. So, for example, how do you do that? Another way, one way of obtaining pure culture is by doing a straight plate method. Para saan to? Itong straight plate, basically, ilalagay mo siya sa petri dish na may agar, i-straight mo siya para ikalat yung cell. Ang purpose is for you to have isolated colonies. Kagaya nitong plate na to, itong at the right, bottom right corner, no, may parang reddish na color ng plate. Merong makapal na growth ng cell, pero at the end, dito banda sa gilid, here, meron kang isolated na colonies. Parang nakahiwa-hiwalay siya na parang dots. Those are isolated colonies. So, pwede kang mag-touch ng isang colony lang doon. At yun ang i-transfer mo sa iyong sterile na medium so that you can have a pure culture. Iisang species lang. Yan. So, yan yung straight method o straight plate method. You can also grow microorganisms. Actually, hindi lang straight plate method. Marami pang iba. Pwede yung dilution technique. Pwede din yung spread plate technique. And when you grow, you need to find the suitable or the optimum environmental requirements for the microorganism that you are culturing para mas mabilis silang grow, mas mag-stay sila with you para ma-enumerate mo sila. Okay. So, if you transfer microorganism in new culture, in a new sterile culture medium, the microorganisms will undergo the stages in the bacterial growth curve. So, yan. Sa umpisa, pag introduce mo a few cells only, they are not yet dividing. So, ang tawag natin sa phase na yan is lag phase. Lag pa siya. It doesn't increase in number, but they increase in size. In preparation for the exponential phase or the log phase, wherein they rapidly increase in number and almost exponentially, they double every generation time until such time that the culture medium will become saturated, the culture medium will start to deplete, and then microorganisms will not increase in number anymore, and we call that phase stationary phase. Okay, from the stationary phase, when toxins, the toxic waste produced by microorganisms accumulate, the food deplete, hindi na siya enough for all of them to survive, the cells will start to die. Those which cannot survive will die. So, tawag natin doon ay death phase or decline phase. Hanggang sa maubos yan sila at maubos din ang iyong culture medium. That is why in the laboratory, if you want to maintain a culture, you need to subculture every now and then. Huwag mong intayin that you reach the phase of prolonged decline before you do that so that you can still save the culture that you want to save, you want to retain, especially if you are studying it for particular characteristics. Next, what are the environmental factors that influence microbial growth? Papasadahan ko lang to, mabilis lang. No? Environmental factors such as temperature, the atmosphere, kasama na dyan yung oxygen requirement, pH, water availability, salinity, pwede din yan, and other requirements. So, for temperature, microorganisms can be classified according to their temperature requirements. So, psychrophile, they love cold environments. So, ito yung mga microorganisms that you will see in the North Pole, in cold environments, doon sa may mga icebergs, in the Arctic, Antarctic regions. Meron din tayong tinatawag na psychotroph. Sila yung important in food spoilage kasi they grow around 20 to 30. But sometimes, class, itong psychotroph, psychrophile yan sila, but they can withstand higher temperature. And they sometimes overlap with the mesophile, wherein mesophile can grow between 25 to 45 degrees centigrade, and most pathogenic microorganisms are in this group. Kasi nga, if they are pathogenic microorganisms of human being, what's the temperature of our body? 37 degrees centigrade, right? So yan ay very optimum for microorganisms. So nag-grow sila dyan. And then, thermophiles, they can live in temperatures higher than 45 degrees centigrade up to 70 degrees centigrade, no, sa mga hot springs. We were able to isolate a thermophile from a certain hot spring in El Nido, Palawan. But higher than 70, pwede nang pangkapi-kape. Higher than 70 degrees centigrade, 70 to 110, that's hyperthermophiles. They are usually the members of the archaea, the microorganisms which can grow in extreme environments, such as the hydrothermal vents. Now, I was very actually interested in isolating a bacterial species that can grow in a volcano, the active volcano. Yan, di ba? Sounds interesting. Oxygen requirement. So, microorganisms can either grow in the presence or absence of oxygen. When they grow only in the presence of oxygen and they will die without it, we call them obligate aerobe. Obligatory aerobic. No, dito. So, ito nga yung example natin dito sa picture, no? Yung agar dito was boiled to drive off oxygen, tapos i-cool down siya, naglagay kayo ng microorganisms. So, anong i-expect mo sa microorganisms? Kapag ka obligate aerobic siya, sa taas yan mag-grow. No, dito. Kasi, syempre, they need to access oxygen. Pero kung facultative anaerobe or facultative aerobe, they can grow kahit saan. May oxygen man yan o wala, they can grow. So, they are well distributed in the culture medium. And, yung obligate anaerobe naman nakikita mo, nandun sila sa baba ng culture medium kasi they are avoiding oxygen. They will die in the presence of oxygen. Ang microaerophile naman, doon din sila sa taas. So, they grow in small amounts of oxygen. So, kailangan din nila ng oxygen. Ang aerotolerant naman, parang facultative aerobe or anaerobe din yan. Kasi, pag sinabi mong aerotolerant, they do not use oxygen. But, they can grow in the presence of it. So, deadma lang, diba? They can coexist. What about pH? Mostly, microorganisms are neutrophiles. Meaning, doon sila sa may neutral banda, near pH 7, ranging from 5 to 8. Something like that. And food can actually be preserved by increasing the acidity, except that some microorganisms really love acid, kagaya ng mga lactobacillus. You know why when you do something, halimbawa, ano bang magandang example? O kimchi, kumakain ba kayo ng kimchi? When I make kimchi, mapapansin mo na sa umpisa, hindi siya maasim. But if you leave it for like 2 to 3 days, eventually, it will become a little sour, which means na nag-decrease ang pH. It means nag-increase yung kanyang acidity. O, tama yun, di ba? Decrease in pH means increase in acidity. Naging mas acidic siya. Kasi that is because of mostly the action of lactic acid bacteria. Ayun, lactic acid bacteria. But in our stomach, the pH of our stomach is actually approximately 2. The Helicobacter pylori grows in our stomach, and that can cause sometimes disease there, yung tinatawag natin na peptic ulcer caused by Helicobacter pylori. Acidophiles and alkaliphiles or alkalophiles. Pag acidophiles, they love acid. Pag alkalophiles or alkaliphiles, they love basic environment. Yeah, so pH. And then we also have the culture media na necessary in growing microorganisms. It can be complex or chemically defined culture media. So ano-ano yun? Bigyan ko kayo ng example here. Ito yung mga karamihan nakikita natin. Example, blood agar. Blood agar is a complex medium used routinely in clinical laboratories because this is a differential culture medium that can differentiate other organisms na pathogenic. Chocolate agar, ayan, that Sounds masarap na. Pati pala microorganisms gusto din ng chocolate. Glucose salts, Maconki agar, nutrient agar, Thayer Martin agar. So, hundreds of types of culture media are available. In our laboratory, sayang no, hindi tayo nakapag face-to-face, makapag-lab sana. In our laboratory, meron tayong available na culture media. Itong Maconki, meron tayo. Meron tayong eosine methylene blue agar, meron tayong TCBS para sa Vibrio, meron din tayong SS, yung Salmonella Shigella agar. So, pwedeng mag-isolate ng microorganisms. So, you can also detect microorganisms na pathogenic in food. Halimbawa, gusto mong mag-conduct ng study ng pathogenic microorganisms present in street food. So, pag nalaman mong meron palang salmonella sa street food, ewan ko na lang kung kakain ka uli ng street food. Pero minsan patibayan lang yan ng sikmura. Wag lang maka-encounter ng HEPA virus sa street food. So, pag sinabi natin class na culture media, ano bang nangyayari dyan? So, for example, we have a selective or differential culture media. Paano natin malalaman yung gusto natin? Dito na lang, in this culture medium in here, ito this one is eosine methylene blue agar. What you can see here is the green metallic sheen and nucleated colony as characteristic of Escherichia coli on the EMB agar or eosine methylene blue agar. So, selective siya in the sense na hindi tutubo yung ibang bacteria maliban sa mga bacteria na enteric o yung kagaya ng salmonella, kagaya ng E. coli, mga enteric bacteria. Sila mag-grow sila dito. Pero, differential din siya in the sense na i-highlight niya yung iyong E. coli. For example, nucleated siya, green siya. Pero kung salmonella yan, class, makikita mo na pinkish ang color niya. Ayan. So, malalaman mo na, salmonella yun pag nag-grow siya doon sa iyong EMB agar, showing the characteristics. Ayan. And then, we have, nabanggit ko na tong atmospheric condition, the aerobic, microaerophilic. Pag nagko-culture tayo, we need to provide atmospheric conditions. So, in other laboratories, mayroon talaga silang mga chamber class. Yung chamber na magpo-provide ng either oxygen o magtatanggal ng oxygen as required by microorganism. Nakakaingit, no? May mga chamber silang ganyan. But eventually, maybe in the future, we can have also this. So, if you become successful in the future and you are multi-millionaire, please come back to the College of Fisheries and Aquatic Sciences and donate a chamber like this. Or baka pwede electron microscope para marami ang pwedeng mag-benefit. Kasi we do not have that yet in whole Palawan. Sana mauna ang WP yung magkaroon ng electron microscope, no? Ayan. So, enrichment culture. Ang enrichment culture naman, class, ang simpleng explanation lang dito is gagayahin niya lang yung environment. So, kung gusto mong mag-culture ng blood bacteria, kailangan may blood ang iyong culture medium. Kung gusto mo naman mag-culture ng plant bacteria, edi dapat may plant extract. Kung soil bacteria, edi dapat may soil extract. So, ganun lang kasimple basta enrichment culture. You enrich your available culture medium with what is required by microorganism. Next. So, what are the methods to detect or measure microbial growth? So, pwede yung direct cell counts. Using microscope na pwedeng mag-count. Pwede din ang tinatawag na cell counting instruments, kagaya ng Coulter counter, flow cytometer. Pwede din, class, i-grow mo muna siya sa culture medium, sa agar, tapos bilangin mo yung colony. Pwede din yung ganon. And then, pwede din siya na, ito na nga yung sinasabi ko, viable cell count. So, you can do plate counts. Gaya dito. Dapat minimum sa isang Petri dish, hindi lalampas ng 300 para pwede pa rin siyang ma-count. Pero kung mga 300 or more yan, hindi na yan siya countable. That means colonies will be overlapping. So, kailangan i-dilute mo siya. So, basta pwedeng i-grow sila on culture medium so you can count the number of cells. Ang tawag natin doon ay viable cell counts. Pwede din, ito class yung sinasabi ko kanina. This is a spread plate method. So you can spread the colonies so that they appear on the surface a little apart from each other so you can easily count, or you can easily isolate as whatever you need to do. Methods to detect or measure microbial growth in with the use of filtration. Yung membrane filtration class, ginagamit natin to kung halimbawa you wanna measure microorganisms in 1 liter of water, for example. So, yung 1 liter of water, kung magsasample ka, i-filter mo siya. Tapos, kung ilan yung na-trap sa filter, you will grow it, and then you can count. Diba? So, pwedeng gawin yan. Like, if you wanna know how many microorganisms are present in 1 liter of drinking water or tap water, pwede mong gawin ang membrane filtration. Next is most probable number. You know this already because this is the method that we use for bacteriological analysis of water. One of the activities that I gave you last time was the making a lab report on bacteriological analysis of water. So most probable number is what we use when we do multiple tube fermentation technique. Another is with the use of spectrophotometer. We also have like this in WPU. Hindi lang nga lang naka-display, but we have spectrophotometer. You can measure the turbidity. Somehow, by doing that, you can count the number of cells in a cell suspension. Okay. Now, let's proceed to microorganisms found in food. Food safety is a global issue. So many microorganisms are present that can cause or spoil food. Sometimes it makes me wonder how the scavengers are surviving. Sinabi ko scavengers, diba? There are people who survive in the leftovers na tinatapon sa basurahan, kinukuha nila, kinakain, pinapagpag. Pero that's full of microorganisms, tapos na-spoil na. Ang problema kasi, sometimes with cooking, you can kill the viable microorganisms. Patay na, wala ng microorganisms. But, the toxin cannot be removed by hot temperature or by heat. So, it's very dangerous pa rin. But sometimes, I guess, napakaganda pag ang immune system na talaga ng tao ang na-develop. No? That's your number one line of defense. Contamination by unsanitary handling of food. Kaya minsan nagkakaroon ng food poisoning. Improper storage and preparation procedure. No? Contamination pa din yan. Imports of fruits and vegetables. Sometimes can bring diseases and parasites. Kaya nga minsan kami parang hindi na makakain ng apple na hindi binabalatan kasi sometimes I'm thinking na bakit kaya to mga ito hindi sila agad-agad nasisira. Maybe some people are using some preservatives, chemical preservatives applied on the fruit so that they would last long. So you really have to remove the peel sometimes, and also if not, you get something to wash it that you can remove the parang yung oily na surface. And ito, even during this time of pandemic, lagi nyo talaga ito mai-encounter. Wash hands properly. That will save you a lot. The transfer of organism from one to another, you transferring the contaminant from the food that you eat and you put in your mouth. So wash your hands properly, especially after the use of toilet. Food production. So we have microorganisms in bread and dairy. In bread, we have the Saccharomyces cerevisiae. Yan yung ating baker's yeast to leaven bread, to ferment, to make alcohol by fermentation. Yung beer, pag gumagawa ng beer, we also use Saccharomyces carlsbergensis and Saccharomyces cerevisiae. Making wine as well. Kung meron nga lang tayong time, di ba? Kung may lang tayong face-to-face, one of the activities that we usually do is to make wine. And then dairy products like the Streptococcus cremoris. So, pwede siyang mag-ferment ng milk and allowed to. So, pwede din yung mga leuconostoc. Kagaya nitong ating mga fermented milk or kasama na din yung ating yogurt. Who likes eating yogurt? Oh, I like yogurt. When I was studying in Japan before, I make my own yogurt. Oh, 'di ba? Kasi nga masarap siya, tas haluan mo siya ng strawberry, haluan mo ng blueberry, slicean mo ng apple, ng kiwi, o 'di ba? Ang sarap na merienda kahit kumain ka ng sangkaterba, no worries for calories. Tapos nakakaganda pa daw siya ng skin. Sabi nung isang Japanese friend ko na matanda na kasi dito yun siya pinanganak sa Pilipinas. Sabi niya yung sa yogurt nga daw pag nag-produce yung parang liquid niya sa taas, pwede daw yun iganyan-ganyan sa mukha para maganda. Anti-aging. Okay. Next, we also have fermented milk beverages, no? Sikat sa atin yung Yakult. Kahit sa palengke, minsan may bebenta ng Yakult. Yan, acidophilus milk. Lactobacillus ang mga nandyan parati, either acidophilus or bulgaricus. Streptococcus lactis, pwede rin. Basta yan ay mga lactic acid bacteria. And also cheese. So, sinong gusto ng cheese? Ilang klase ng cheeses ang alam ninyo? Baka mamaya yung alam lang natin na cheese, yung Eden cheese, yung cheesy, yung Dane, ano pa yan? Iisang kulay lang nila lahat, parang yellow lahat ang cheese natin dito. Na maraming kulay kaya ang cheese. May black cheese, isa sa mga mahal ang blue cheese, yung white, yellow. Yun pa lang so far naman na natikman ko. Nung makita ko yung Australian friend ko na kumakain ng black cheese, parang lagay niya sa tinapay, parang sarap na sarap talaga siya. Wow, yummy. And I was wondering, sana makakain din ako niyan. Nung natikman ko, my goodness. Sige nang, kahit hindi ko na matikman uli. Ganon. And then, so cheese. Ang cheese is galing din siya sa milk and that is produced by bacteria. They form a curd. Oh, parang curdling yung process niya. So, meron mga factories na talaga namang pangmaramihan, pangmalakasan no, pag maggawa ng cheeses. Because the cheese industry is a big industry because a lot of people are using cheese. Diba? Pag mag-queso de bola, pagka-Christmas, kahit mahal na mahal, binibili. Pag kumain ka ng spaghetti, may cheese. Gumawa ka ng fruit salad, may cheese. Pagka sa mga countries pa naman na ano ba to, kagaya ng Italy, di ba? They like pasta so much and pasta usually comes with cheese. Ay, nakakapurga. Okay, I have a question here. Can you give examples of microbial products we usually buy as food? I want to see your answer in our chat box. Can you give examples of microbial products we usually buy as food? Ano ba sa tingin nyo yung, marami na rin ako nabanggit kanina, but apart doon sa mga nabanggit ko, what are other microbial products that we have and we usually buy as food? Sige nga, tingnan ko nga yung mga sagot ninyo. Okay, any answer from you? Give an example. Okay, so I will see your answers anyway. So here are other products that are produced by microorganisms. First, vinegar. So this is ethyl alcohol or acetic acid produced by Acetobacter aceti. Pickles, olives, soy sauce, soy products, fermented meats, alcoholic beverages, and nata de coco. Hey, I'm so happy kasi my friend found a mother liquor o yung starter solution ng nata de coco and it's arriving soon. It's already coming on its way. I'm gonna be able to make nata de coco. Patitikmin ko kayo pag nakapasok na kayo ha. Gagawa tayo ng nata de coco. I really like nata de coco. And one good thing is that when I went to the market, nakita ko na nasasayang lang lahat doon yung sabaw ng nyog. And that can be transformed, hindi. That can be used as a source of nata de coco. So, pwedeng-pwedeng gawin. O kung kayo may mga nyugan kayo, pag nagkukopras kayo, ay nako, sabihin ko sa inyo, Baka yayaman tayo sa data de coco. Hindi, joke lang. Okay, so tignan ko nga. Sabi ni Maricar, wine. Bakit may question mark? Yes, wine. Kimchi. Fermented fish. Bagoong ba yan, Christine? Oo, bagoong. That's a product of microorganisms. Vinegar, that's correct, Arnel. Si Maricar, kalburong mangga, ma'am. You like kalburong mangga. Oh. Fermented fish. Soy sauce, that's correct. Yakult, nata de coco, beer, beverages, beer, wine. Okay. Thank you for participating. You are all correct. Tama yan. So, ayan we have a process here of making red wine. And, excited sana ako ipagawa sa inyo to Pero actually, pwede talaga. Hindi ko lang siya hindi lang nga natin kasi masyadong inabot yung biotechnology. But I could have asked you to make wine even at home because it is doable. Wag naman galing sa grapes kasi very expensive. But maybe you can make a wine out of watermelon kasi maraming katas. Pineapple that I like. It smells really, really good. Okay? Do you have any question, class, with this part? Itong part lang natin. If you have question, please type in our chat box. Before I proceed to microbial genetics. Question. Do you have any question? Bread, Krisa. Oh, totoo, bread. Bakit tumawa si Joman? Saan ang sagot? Beverages beer. Okay, that's correct. Ayan. Nasaan kaya yung ating ibang mga kaklase, kaklase natin? There are only 48 watching. 3 sections kayo. Oh, mag-final exam sila. I like. Sige lang. Let's find an alternative for them. We need to help each other. So next, central dogma. When the DNA is replicated and the DNA is transcribed to RNA, and RNA is used to make protein, then we call that the central dogma of life. And that's the same with microorganisms. So, ano lang to, mabilisan lang kasi we already have this and you can even replay this in one of the lectures that we have before. So, mabilis lang. Ito yung ating DNA. We have alternating sugar, phosphate, and meron din tayong nucleotides. Thymine paired with adenine, guanine paired with cytosine. Thymine and adenine has 2 hydrogen bonds. Guanine and cytosine, they have 3 hydrogen bonds. Ang difference between RNA and DNA when it comes to structure is that the DNA is double-stranded, double helix, and RNA is single. When it comes to components in the nucleobases or the nucleotides, RNA has cytosine, guanine, adenine, and uracil. Remember that, uracil. But the DNA do have cytosine, guanine, adenine, and thymine. Wala siyang uracil. RNA doesn't have thymine but has uracil. DNA doesn't have uracil but has thymine. Okay, so para ma-refresh tayo kung ano ang replication, we have to discuss it again. So, this is an overview of replication. You have a double-stranded DNA cleaved by an enzyme, a polymerase, RNA polymerase. Both strands are now open for replication. One strand is having a continuous replication and the other one has a discontinuous replication. So if we look closer into each of these strands, tingnan lang muna natin si leading strand muna tayo. Pag sinabi kong leading strand, ito class yung continuous replication. So what happens to the leading strand? Again, when the DNA is cleaved by an enzyme, a strand becomes open as template, and there is a continuous replication from one end to the other end until it is completely done. Walang putol yan. No? Basta leading strand. Yan ang tatandaan ninyo. And in the replication, meron ding lagging strand. Sa lagging strand naman, leading strand, lagging strand, ito. Lagging strand, ano naman ang nangyayari? So, it's just the same, no? The DNA is cleaved, it becomes an open template, but in the lagging strand, the replication is not continuous o putol-putol. May isang fragment dito, another fragment na naman, and then another fragment. Tapos, there will be another enzyme na magre-repair ng gap para makumpleto pa din siya. Yung ating mga DNA ligases, no, nire-repair yan nila para maging kompleto. Ang tawag natin doon sa mga maliliit na fragments na yun ay ano nga? Okazaki fragment, no? That's coming from a Japanese yung nag-discover niya, no? Okazaki fragment. So, yan ang lagging strand. Basta hindi siya tuloy-tuloy na ginawa, discontinuous siya, that is lagging strand. Now, let's go to transcription. Okay. Nang pinakita ko ang transcription sa inyo, dati-dati hindi natin nakita siya as like really animated. Oh, by the way, ulitin ko lang dito sa replication, it will always follow the base pairing rule. A with T and C with G. Dito sa transcription now, remember that A will be paired with U. So this is a DNA template. Now you can see that the base pairing rule is there, G with C, here, G with C, D with A, C with G, A with T, and so on and so forth. So this DNA will have the promoter region. Now you have the coding region, the promoter site, and the terminal site. So the enzyme that will cleave the DNA will bind at the promoter site and will cleave or will unwind our DNA. So you have now the bubble template. Ayan, para siyang bubble na template. And then, based on the base pairing rule, A with U, G with C, T with A, yan. Yung pink na yan na nabubuo, this one is what we call the messenger RNA. So, transcription, pag sinabi mong transcription, the byproduct is RNA. Okay. So, when transcription is finished, the DNA will just be restored back into its original state. Then, the RNA will be detached and it will be deciphered. So, this is the RNA that will be read in order to do protein synthesis. Okay. So, if we look at it closely, makikita natin na itong gene na to na sinasabi dito, this is our coding region. The promoter region and the coding region. So the base sequence of the promoter directs the enzyme RNA polymerase to bind to the DNA to initiate transcription. Pag sinabi nating initiate, ibig sabihin nun, i-unwind niya yung iyong DNA so that the strand will be open as template. So reading that, if we have to look at it closely, yan nakikita niyo yung base pairing natin, diba? You have A, T, U, A, baliktad pala, galing tayo sa blue. T with A with U, C with G, and so on. So ito nga, yung tatlong natitirang nucleotides here, we have A, T, and A. So the pair of A is U, the pair of T is A, and the pair of A again is U. So yun lang pinagkaiba niya sa DNA, ang uracil. And then, when the last stretch of messenger RNA is read, it will be released. So this completes our transcription. Okay. Pero Siyempre, pagkatapos nito, ito hindi ko to nabanggit sa inyo dati, hindi naman siya agad-agad class na kung ano yung nandyan sa messenger RNA niyan, diretso na yan siya kaagad. Kasi nga, meron tayong tinatawag na coding region and non-coding region. Ang tawag natin doon ay the exon and the introns. Introns are not coding region or non-coding region. They are not protein building regions. Wala silang protein building instructions. So most cells or most eukaryotic genes contain introns. They are sequences that do not contain protein building instructions. The introns sit between exons. So kailangan siyang i-excise. Kailangan siyang tanggalin kasi pampagulo siya dyan. So the pre-mRNA is modified. Anong minomodify sa kanya? Tinatanggal ang intron. And then, lalagyan siya ng cap so that it will help not to have any more nuisance, yung mga pangpagulo. The length of the tail determines. Okay? So, ano ngayon? Sabi niya, the length of, so a long tail of hundreds of adenines. Ang ilalagay niya doon sa dulo, the tail determine how long the mature messenger RNA will last. Yan. So, meron ka nang tinanggal niya yung intron at pinagdugtong niya. So, ito lang yung coding na region. Yan lang ang merong code na matatranscribe natin to protein. Ang tawag natin dyan is mature messenger RNA. Ready for coding. So, how do we do that? Of course, we need to read the code. And that is by using our genetic code. Ito yung ating codon, genetic table. So, ang codon natin, we have a combination of either U, C, A, and G. So, ito yung ating mga amino acids. So, for example, if we get a combination of natin ha, U, C, and A, that is serine. So, pag yan ang bit-bit ng transfer RNA, dadalhin niya ang serine. Pag CAG, glutamine. Tapos, when you have AGU, that would give you another serine. Okay, what about ano pa ba? Halimbawa, A, C, U. That's threonine. Okay, so, dito sa ating codon class, meron din tayo ditong tinatawag na stop codon. So, meron tayong tatlong stop codon. That is UAA, UAA, tawag ko nga dyan para maalala ko eh. UAA, UAG, UWAG, UAA, and then the UGA, yung UGA. There are 3 stop codons. So, pag na-load yung stop codon, tapos na rin ang protein synthesis. So, what else are participating in protein synthesis? Meron tayong RNAs, translation RNAs, transfer RNA, ribosomal RNA, and the messenger RNA. So, ano ang ganap nila? What are the uses of these RNAs in the process of translation? Pag sinabi natin translation, that's protein synthesis, right? So, in protein synthesis, translation begins when an initiator tRNA is loaded into the platform of small ribosomal subunit. And then, next, the messenger RNA will come. So, messenger RNA carries the code, right? That was, we showed you that earlier. And then, another one comes, large ribosomal subunit. So, these 4 together, we call this the initiation complex. And then, we have in here a simplified model of the binding sites. You have the binding site for the messenger RNA and the binding site for here the transfer RNAs. So, the first and the second transfer RNA. Reading the code in the messenger RNA, transfer RNA will bring the anticodon. And that carries the amino acid. Okay, and then another transfer RNA will come reading the next sequence of amino acid. Now, sequence of the code in the messenger RNA. And then, now you have two amino acids, and these two amino acids will form a polypeptide chain. Yan, para dikit-dikit sila. Siyempre, kung hindi naka-chain yan sila, then hiwa-hiwalay, amino acid pa rin sila. But if you put them together, they will become eventually protein. So, ma-dislodge yung initiator transfer RNA. The next transfer RNA will come carrying another amino acid until this process will continue until such time that you have a fully grown polypeptide chain. But when the stop codon is loaded into the platform, your translation process is terminated. So now your translation is complete with a long chain of amino acid here forming protein. Remember that amino acids are the building blocks of protein. Did you get that? Okay. Now, if you have question, if you have question naman regarding bacterial genetics, please post, and I will discuss that later. So let me check again. If you have questions, none. I would assume na intindihan nyo ang lecture kasi no question eh. Minsan pa naman pag no question, dalawang bagay lang. Pwedeng naintindihan, pwede ring hindi naintindihan. Would just assume na niintindihan nyo. Grabe na ang effort diba ni ma'am? Ano na to? A for the effort. Sige. I will just have a look later kung meron kayong tanong in bacterial genetics. Now, I would like to go into microbes in the environment. So, microbes are essential for life on Earth. Microorganisms function in photosynthesis. Meron tayong mga microalgae, meron tayong mga photosynthetic bacteria. So that means that they are very important in the food chain. And of course, alam na natin, 'di ba? Nung mga elementary tayo, high school tayo, pag sinabing bacteria, ang alam lang natin, decomposer silang lahat. We never know that sa food chain, 'di ba? Ano bang series ng food chain? Producer, primary consumer, secondary consumer, ano pa yan, tertiary consumer, decomposer. Pag sinabi mong bacteria, ilagay na nila yan lahat, decomposer yan. But that's a no-no. Bacteria, microorganisms can actually occupy all trophic level. Producer siya kasi photosynthetic. Primary consumer siya kasi siya ay pathogens din ng plant. Secondary consumer siya kasi siya ay pathogens ng primary consumer. Tertiary consumer siya kasi siya ay pathogen ng secondary consumer. So, microorganisms occupy all trophic levels. Huwag niyong kakalimutan, but they are very important in the composition process. Ma-imagine mo kung walang microorganisms, pwede nating itabi yung ating mga mahal sa buhay. Paitabi mo lang, di ba? Hindi mabubulok yan. Hindi babaho. Bumili ka ng isda kahapon, kahit next week mo pagalawin, walang mangyayaring pag walang microorganism. Anyway, and of course, nitrogen fixation. Some bacteria can take nitrogen from air and incorporate it into the soil so that it will help the plants. Plants cannot acquire the atmospheric nitrogen kasi strongly bound yan. Yung N2, tatlong bonds ang meron yan. So, hindi yan nila nagagamit. Kailangan siyang i-nitrogen fix ng microorganisms before plants can use it. Microorganisms are also beneficial in digestion in animals. Kaya nga, di ba, meron tayong mga iniinom na probiotics. That's good for our tummies. And also for animals, kagaya ng ruminants, itong mga cow, carabao, ganyan. Bakit sila nakakadigest ng grass? Diba tayo, kung makakadigest tayo ng grass and then we can just go grazing. Huwag na tayo magluto. Diretso na tayong kumain ng damo. But we do not have cellulase, so we cannot digest cellulose. Animals like goats, cows, they have microorganisms that produce cellulase for them. Kaya nakakadigest sila ng cellulose from plants. Vitamin K and B synthesis in humans. So, wag kayong nagagalit pag umuutot kayo. Pag umuutot kayo minsan, those are microorganisms present in the large intestines. But mind you, they produce vitamin K for you. Vitamin K is very important for blood clotting. Pag wala kayo niyan, konting sugat lang, magdudugo, pwede magkaroon ng hemorrhage. Ayan. So in medicine, many antibiotics and other drugs are naturally synthesized by microbes. Kagaya ng discovery ni Alexander Fleming, the penicillin from Penicillium fungus. So, meron ng penicillin at marami pang iba, no? And then, important and beneficial biological functions of microbes in the food industry. Ito na yung mga sinabi ko kanina, kung ano ba yung mga possible food. Ang mamahal nito alcoholic beverages. My God, yung mga seasoned wine. Magkano ang isang bote? Don't you know na merong wine na million ang halaga? Yung mga parang century wine, mga ganun. Tapos talaga super, super mahal. Other people, the rich families would have a cellar. May mga collection sila ng mga wine. Parang magandang magkaroon ng ganun, no? Bread, vinegar, soy sauce, cheese, pickles, olives, sauerkraut. Itong sauerkraut ay ano lang to, parang pickled cabbage. Yogurt, buttermilk, sour cream, coffee, chocolate. Yung coffee na fermented. Chocolate, yung pinakamaganda is talagang product ng fermentation, hindi yung niluto-luto lang. Ham, sausages. Ayan. Another important and beneficial biological function of microbes is genetic engineering. With the advances in gene splicing, there is now this so-called recombinant microbes that they can use in order to address several problems. Kagaya halimbawa ng dwarfism, no? Yung human growth hormone na pwede nang mapalitan. When the gene is not capable of producing insulin, that can cause diabetes, that can also be done through gene therapy. And microorganisms are being used in there. Blood clotting factor, recombinant vaccine, ayan. Ang example natin nga dito, hepatitis A and B. Pero, syempre, we cannot forget COVID. It's very, very crucial nowadays that vaccines be purchased so that people will be vaccinated and will have herd immunity. Para hindi na magkaroon ng transmission, makabalik na tayo sa dating normal. Human hemoglobin, taxol, erythropoietin, monoclonal antibodies. And so these are the things that can use genetic engineering to be achieved using microorganisms. Medical research. So, microorganisms are well suited for biological and medical research for several reasons. Kasi they are so easy to culture, short generation time, genetic material nila is a little simpler, and easily manipulated. Now, if you remember what I asked you to give reflection, the biotechnology of microorganism that can produce antibiotic. That's an example of medical research using microorganisms. So you bio majors, you can actually choose to do research related to this. And then, microbes and diseases. So, aroy, ang daming emerging diseases talaga. No, we have now war against infectious diseases. We are now in current pandemic. So, we are in a war. War against the virus. Pero alam nyo ba tong mga virus na to, itong mga bacteria na to, they can even be used as biological weapon. Oh, my conspiracy theory. What do you think of the COVID-19? Meron ba tong hidden agenda? But, emerging diseases. So, kagaya nga ng COVID-19. Yung AIDS, nako, in the Philippines, Puerto Princesa is one of the highest rating when it comes to AIDS. Hantavirus, Ebola fever, Lyme disease, hepatitis. Karamihan dito, class, these are viral, no? Antibiotic and drug resistance. Marami na, like tuberculosis, gonorrhea. Ang malaria, dito sa atin, very common din yan. But malaria is not really caused by bacteria or virus, ha? I just would like to emphasize that malaria is caused by a parasite. Plasmodium, that's a parasite. A member of Protist group. Sila ay protist. Parasite yan sila. Yung apat, Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, and Plasmodium ovale. Yan, apat na common types of malaria here in the Philippines and here in Palawan. Today, infectious diseases cause 50% of the 52 million worldwide deaths per year. So, yung kalahati nun, ng 52 million na namamatay taon-taon, pwedeng hypertension, diabetes, yung mga ganon. So, pero kalahati talaga, no, by infectious diseases. So, next, what are those? Examples, no? The acute respiratory, bacterial or viral, namamatay, mga nasa 4 million. Diarrheal diseases, tuberculosis, malaria, hepatitis, measles, AIDS, neonatal tetanus. Ang dami. So, a total of mga nasa 50 plus million. No? So, scary. Example nga ito, oh. This is a neonatal tetanus that kills over 600 infants every year, all over the world. So, mahirap. No? Kaya, talagang for pregnant women, they have to be taken care by their OB-GYN para hindi magkaroon ng mga ganito. Kaya talagang, ako nung I got pregnant with my baby, I got the tetanus toxoid vaccine para maiwasan ito. Microbes and disease in human history. Oh, ito kasi history, pag titingnan natin ngayon, no, ditong pandemic na to, no, it's killing hundreds of thousands of people all over the world. But in the history, there were more diseases, microbial diseases that killed millions of people. For example, we have the bubonic plague or the Black Death. Several devastating epidemics throughout history. High mortality up to 80% of those infected. Alam niyo ba yung expression ng British? Sana walang nanonood na British. When you sneeze and they say bless you, my friend before told me that that's because of the plague that happened in the Europe before. Ayaw ko kung Britain na nga ba. Kasi the first symptom actually that you got infected is sneezing. So when you sneeze, hatchoo! Ganyan, sabihin nila, bless you. That means bless your soul because you are going to die. Na? Ang sakit naman. Pero naging expression na yun until now. Pagka nag-sneeze ka, yung mga nakakaalam na, they will say, bless you. But I think it's genuinely wanting to bless you, but not really to say bless your soul because you're going to die. Hindi naman, no? So, in 1347 to 1351, over 75 million died in Europe, in Asia, and Africa. 25% of population ng Europe namatay. Diyos ko. That's because of this plague. The cause was unknown, imagine, but it's a bacterial disease transmitted by a rat flea. Kaya mag-ingat kayo sa mga ano nga yan, yung flea lang, bitik lang pero nakakamatay. Sige, example niyan swollen lymph nodes in bubonic plague infection. Okay, so paano ba nag-spread yan sila? Basta pag sinabi nating worldwide, that affects mostly the continents, no, different continents in the world. Smallpox. One of the deadliest human infectious diseases in the history was actually the smallpox virus. The first known case was in 1175 BC when the Egyptian pharaoh Ramses V died from smallpox. Yeah. Several 100 million deaths through history, up to 90% of the Native American population was killed 10% lang ang natira. By smallpox and other diseases like measles and plague. All of these are viral. Introduced during European conquest. This is a biological weapon. Yan yung sinabi ko sa inyo kanina. Native population of Central and South America dropped from 130 to 1.6 million over several decades because of smallpox. And that was a biological weapon. Oh, wala naman sanang no. Naka-live ba tayo? Baka mamaya meron na magdampot sa akin at bakit ko sinasabing biological weapon. But that's recorded in the history. 600,000 deaths per year. That's from 1500 to 1700. Diyos ko, ilang taon yan. Tapos 600,000 per year. Naubos sila. That's, I'm just smiling. But, when we look at it, when we reflect, talagang it's really devastating. And let's pray that this will not happen again. Even with this pandemic that we are facing now, we sometimes, I could always thank the advances in science, kasi mabilis na yung tracing, mabilis na yung paggawa ng vaccine. Dati, bago ka makagawa ng vaccine, tag 10 taon. But now, wala pang 1 taon, may vaccine na sila, diba? So, the advances of science can also be good. And smallpox infection. Tingnan mo naman, kawawang kawawa. The smallpox was eradicated completely in 1977 when Edward Jenner developed the vaccine. So see, even in the history, vaccine saved lives. Dati-dati pa yan. No? Ngayon, we are also holding on to vaccine. Nag-aaway na yung mga tao kasi hindi pa nakabili ng vaccine and mga political will, political agenda. Tuberculosis also is one of the leading causes of death before, pero ngayon libre pati ang libre ang diagnosis, libre ang treatment ng tuberculosis. Punta ka lang sa tb.doon sa halimbawa sa ONP, meron yan sila. Caused by a bacterium. Ano ba mga bacteria na nagko-cause nito? Meron na tayo nito sa list niyo. You have Mycobacterium tuberculosis, Streptococcus tubercle bacilli. Yan, mga causative agent ng tuberculosis. But, there's one thing. When I got sick in 2019, I was hoping na yung bukol ko ay hindi cancer, but only tuberculosis. Talaga naman, hindi nangyari. Pero mayroong tinatawag class na extrapulmonary tuberculosis, not affecting the lungs but affecting other parts of the body. Like this, patient with lymph node necrosis. Tuberculosis siya. No? So scary. And then, AIDS. Huwag na natin yan aralin. Are you familiar with acquired immune deficiency syndrome? The AIDS victims or AIDS positive or HIV positive people, ang pinaka-problema sa kanila class is they become so sensitive. They get easily they can easily contact infection. Mabilis silang magkaroon ng mga infection because they now considered immunocompromised. Their immune system is compromised. Yan yung nangyayari with AIDS. At hanggang ngayon, wala pa rin gamot sa HIV. And ang maganda lang sa kanya, hindi siya nakakahawa via direct contact. Until now, sexual contact pa rin siya at saka blood transfusion lang pwedeng makahawa. Look at this. This is an African AIDS patient with slim disease. Buhay pa yan, pero parang butot balat na. Literal na butot balat. And then, another one, extensive symmetric tumor lesion in AIDS patient kasi nga very sensitive na sila. This one is oral candidiasis or yeast infection in AIDS patient. Okay. And then, let us now go into the different routes of infection. Paano nga ba nakakahawa yung mga sakit? Microbial diseases. Okay? So first, we have the respiratory droplets. Yung droplet contact. When someone cough, coughs or sneezes, airborne droplets scattered, and that could be contracted by other people. Just like ang ating COVID-19 ngayon ay nako-contract siya via droplet. That's why we are asked to wear mask and face shield. So, yung COVID-19, tingnan nyo, this is the coverage of the pandemic ng COVID-19. Lahat talaga, walang naligtas. At least, may mga countries na medyo mababa ang infection, pero ang bilis talaga niyang kumalat. Next to droplet contact are fomites. Pag sinabi natin fomites, this one is indirect contact. Indirect means inanimate objects that spread diseases. Kagaya ng kung mag-share kayo ng drinking cups, share kayo ng kutsara, ng plato. Yung mga minsan pag kumakain tayo sa labas, sa restaurant, and they are not washing it properly, they are not disinfecting, especially yung silverware, the spoons and forks. Pag nasa mga restaurants kayo and you see them not sterilizing, you can ask for hot water, put it in a cup, and sterilize your silverware. Kahit sa mga bata, yung mga public playgrounds, pwedeng makakuha sila ng sakit. Next to this, kanina yung indirect, this one is direct body contact. Kagaya ng STD, impetigo. Impetigo ito, yung parang siyang infection na malapit sa bibig. Meron siyang bacterial infection, kagaya nito. So, person-to-person contact. Yung sexually transmitted diseases, person-to-person contact din. Okay, next, we have also fecal-oral contamination. So, feces contamination of food, even dirty hands pag hindi naghugas, galing sa banyo, sa CR. And then, also, yung ating mga pagkain, pag hindi na-cover, gapang-gapangan ng cockroach, patungan ng housefly, ng fruit fly. And then, pwede siyang maghawa, pwede siyang magdala ng sakit. Pag yan, yung mga ipis na yan ay gumapang sa tae, gumapang din sa pagkain mo, gumapang sa toothbrush mo, gumapang sa kutsara mo, sa platuhan mo. Vector-borne. Ito naman yung mga dala-dala siya ng arthropods vectors such as flies, fleas, mosquitoes, tick. Kagaya lang halimbawa class ng malaria, carried by Anopheline mosquito. So, also, yung ating dengue. Dengue fever is a viral infection also carried by mosquito. Sa ibang bansa, meron silang yung mga cheche fly na nagkikarry ng night blindness, sleeping fever, o sleeping, basta may mga ganun silang mga fever. Yan, vector-borne, basta dala siya ng ibang organism. Next, airborne. Ito yung mga contagious diseases, no? Pwede siyang madala ng particles, so pwede siyang sa hangin lang because the bacteria can actually stay for long in the air. Okay. And then, bibilisan ko lang, parenteral or iatrogenic. Ito yung via blood transfusion. Kaya pag nag-donate ng blood, talagang ini-screen nila ng maayos para hindi magkaroon ng hawaan ng sakit. Ito, tetanus. Tinan mo, karpentero, na pukpok niya yung kamay niya. Ayan. Kailangan may karoon kayo ng anti-tetanus kasi it's deadly. Horizontal or vertical. Pwede yan, no? Yung mother to the baby via the umbilical cord. Okay. Now, I would like to go straight to the aquatic kasi our time is almost finished. Mahaba na yung ating oras. Now, first, we have fish and shellfish. So, fresh fish have microbes kagaya ng Salmonella, Vibrio, Clostridium, Cryptococcus, Candida, and they cause spoilage. Kasi fish and shellfish do have soft body. So, they can easily be ma-attack siya kaagad ng microorganisms. So examples of this class ng mga diseases, no, sa pwede magkaroon ng fish and shellfish diseases caused by microorganisms, kagaya lang halimbawa ng vibriosis. Ito siya, sa isda. Hemorrhagic skin lesion, sa dorsal part. So, maraming isda ang pwedeng mamatay with microbial diseases. Ito examples lang to, ha. And then, bacterial gill disease, Plectrophomus leopardus, ayan, ang gill niya ay merong infection. So, kung sino sa inyo ang may mga aquarium na nasa bahay, minsan meron talagang bacterial o microbial diseases, fungal diseases, nangyayari yan sa mga aquarium fishes. And Chromeleptis altivelis infected with Flexibacter maritimus, mga hemorrhagic and eroded caudal and pectoral fins. So, mga ganyan yung characteristics niya. So, marami pang iba. You already have the list of this in your module. Bago ako magtapos, those in the College of Education, later on you will become teachers. You will be able to use microbiology in school, even for high school. There are already a lot of opportunities for researchers in microbiology, and you can do research with your high school students on microorganisms, showing how microorganisms can affect the population, or also how you can control microorganisms, such as study of antimicrobial potential of plant extracts, antimicrobial potential of animal extracts, and things like that. And I would like to encourage all of you to get that book. Mayroon akong link na binigay doon sa aking mga estudyante sa College of Education, the book of microbiology for high school. Now, do you have any question? I think that ends my lecture. Ang haba na, no? Hindi kayo masyadong napagod makinig. Mga 1 oras at kalahati din tayong nag-lecture. Now, I would like to see if you have questions. Let me have a look at our group chat. Yes, merong hidden agenda. Merong hidden agenda ba talaga ang pandemic? Sige, biological warfare, pwede. Bioweapon ng China. Ay, ganon. Mabenta face mask, no, yumaman yung mga gumagawa ng ganon. And other things. So, nakakatakot siya kasi nga, when you know microbiology, the recent class, why I'm teaching you these topics, kasi for you to be able to protect yourself. Your knowledge here, please use it wisely. Bawal yung gumawa ng biological weapon, in the future. Tapos sabihin niyo, sino nagturo sa inyo? Ay, naku, si Ma'am Jo po nagturo sa amin. Patay tayo dyan. Use it wisely. Gusto ko, pwede nyong gamitin yan to produce products that you can do for business, mga ganun. O kaya help people na may mga problema with microbial diseases sa kanilang tanim, sa kanilang mga hayop, but not to harm people. Is that okay? Okay, so I'm waiting if you have questions. Sa China nagsimula, tapos sila din unang nagka-vaccine. Diba? Sometimes we can really be suspecting. Ay, nako. Sige. So, do you have any question? Class, kayo ba merong natutunan sa klase natin? Meron ba kayong natutunan sa microbiology? Our HR in WPU has sent us a link for faculty evaluation. So I'm gonna send it to you in our chat, no, GC, in our GC later, and kindly evaluate me kung according doon sa criteria nila. Okay? So, meron ba kayong natutunan? Okay, this is not part of your requirement class, but if you can, if you are listening to me now, I would like to ask a short reflection sa inyo. Bonus activity siya. So it means not part siya ng total items ng inyong activities, but this is a bonus activity para sa mga gustong ma-flat 1 ang microbiology. I would like to ask the reflection from you. You can submit it in a Google Classroom. Mag-post lang ako ng ng classwork doon. So you can post a reflection. I would like you to evaluate our semester. Magbibigay din ako class ng guide question ko paano i-evaluate. But I would like you also to evaluate our delivery of instruction. Hindi ko kasi alam kung ng ibang ang method ng ibang teacher para magturo sa inyo, but for me, we exhausted our possible online classes through Google Meet, Google Classroom, and through YouTube Live. So please give your honest reflection and evaluation of my teaching method as well, and kung ang pinakauna doon is ano ang pinaka natutunan ninyo sa microbiology class. Isusulat ko yan at ipopost ko lang sa inyong classroom or sa GC. So ano ang pinaka natutunan ninyo? What is your most valuable learning in microbiology? And then how was the delivery of instruction by your instructor? And how can you apply what you have learned? Yun, mga ganun lang na question sa reflection. Bonus yun. Okay? May minimum words, wala. Bahala na kayo kung gusto nyo ng essay na mahabang-mahaba, pwede yan. No? O kung gusto nyo naman ng isa-dalawang sentences lang at nandoon na yung gusto mong sabihin, that's just a bonus activity. So walang format, you can freely say something. So are we good? Okay na ba tayo? Do you have any more pahabol? Any question at all? Last meeting na natin to, class. Yehey ba? Nakasubmit na ba kayo lahat? Hindi ko pa na-check yung mga bagong submissions kung sino na yung mga kompleto at sino yung hindi pa kompleto. Have you submitted all your activities and all requirements? And as I promised, as long as you are tuning in this lecture, no more finals. So ito na yung pinaka-finals natin, and I hope you learned something. Ma'am, may napanood po ako. May positive po ba ang spoils food? Ano spoiled food? Positive effects ng spoiled food? Oh, actually, yung fermentation is ano yan? Spoiling of food. 'Di ba? Pinapabulok mo yung pagkain eh. So that's precisely spoiling of food. Kaya siya nangangasim, kaya ganun. And it's beneficial because microorganisms in there are actually transforming your substrate into something that is more beneficial to the body. So meron talaga siyang meron siyang positive na effect. Kasi yung spoiling of food is also a process of fermentation. Oh, thank you everyone for tuning in. Sige, antay lang ako. Hindi pa muna ako mag-end. Pero yung iba, kung mayroong mga gagawin, pwede na kayong sign out. That's okay. Pero yung iba na gusto pang magtanong, At I will still stay until mga 11:15 para sa mga gustong magtanong. Naninigurado lang ako. Although pwede ko itong ma-replay ang ating video para makita yung mga top chat natin. Naninigurado lang ako na makukuha ko lahat ng inyong mga pangalan. So, nagpi-print screen ako ng inyong mga sagot, ng inyong mga pangalan. Tapos ipopost ko to para masigurado ko na lahat kayo nandito. If you are watching and you cannot type kasi may mga ganong problema, please send me a private message. If you are watching and make sure you do also some screenshots to prove na talagang nanood kayo kasi finals natin ito. Okay? Alright. So, any more question? Baba ko ha. Thank you everyone for being patient. Thank you for participating. And I hope that you learned something. Maraming thank you ako nakikita. Thank you, thank you, thank you. Thank you talaga sa inyo din. Alright. Oo. Uy, baka meron pang hindi nakapag-subscribe. Mag-subscribe na kayo ha. Para sa higher course ninyo, gagamitin pa rin natin itong platform na ito para mag-lecture, mag-post na mga subsidy lectures o mga supplemental lectures. So, if you are not yet subscribed to this channel, please subscribe. Pang-inspire kay ma'am. Pag nakikita kong madami ang nag-subscribe. Thank you! Wala nang magtatanong. O sige lang, you can just leave now if you're done. For those who would like to ask question, you can still post. I'm still tuning in. Thank you po, ma'am. Kayo lang po ang nagdi-discuss sa amin. Ay, ganon! May pa-hit the notification bell din po. Oo, please! Thank you! I like that notification bell. I like the din. Subscribe. We're done? Wala nang gusto magtanong. Keep safe everyone. So, thank you. And do your best to finish all your modules para walang INC sa inyo ngayong semester. Bye-bye. Thank you very much. Bye-bye, classmates. Bye-bye, ma'am. O, sige, bye. Bye, Jovan. Bye, everyone. Sir Palya, if you are there po, still, thank you very much for tuning in. If you are there po, still, thank you very much for tuning in. Okay. So, I'm going to end our streaming now. Wala na rin namang nagtatanong. Oh, advance happy Valentine's. Good luck, everyone. Bye-bye.